Hi, my name is Isabel Garcia, and today I will be explaining our research made with Dr. Ariela Vergara-Jaque and also Dr. Horacio Paulet that is called Identification of Integrins as Pharmacological Targets for the Design of Therapies Against Colorectal Cancer. This research was made at the Center for Bioinformatics and Molecular Simulations at the University of Talca. As a background, colorectal cancer is the third most diagnosed cancer in the world, with very low suspicion of survival if it is not detected at an early stage. In this context, there is a really big necessity to find alternatives that are complementary to surgical and chemotherapy approaches that already exist. The identification of proteins that affect cellular process associated with colorectal cancer is essential to define pharmacological targets that facilitate the binding of medical formulations. In this necessity, we were able to find a family of proteins that is called integrins that are related to fundamental steps for cell reproduction and survival. Various isoforms of those proteins are overexpressed in colorectal cancer, and specifically the integrin called alpha 5 beta 1, alpha B beta 6, and alpha B beta 3 were the ones that we use in this investigation because they were linked directly to colorectal cancer. With that background, the objectives of our investigation were to define a set of 6 peptides, 3 of them with the RGD motif into their sequence and 3 of them with the NGR motif into their sequence, so we can put them into the protein set that we already defined, and the final aim is to find a complex with pharmacological potential. To establish a characterization in a structural level, we follow a computational workflow that was initially a sequence alignment of the subunits that are forming the integrin, the protein, the alpha and the beta subunits. Next to that, we make a geometric analysis with GeoMinder, and we were able to extend the sequence alignment and find patterns that were not only shared into their sequence, but also into the way that the residues are located into the space. Following that, we made a peptide modeling for the set of peptides, and we made molecular coupling or molecular docking with autodock crampep for the 18 combinations that we can establish with 6 peptides and 3 proteins. And based on the results and the alignments of the results, we made molecular dynamic simulations for the peptide that we find that works the best, that is this peptide in B in this image, and also the 3 proteins that we were trying to characterize in our investigation. And finally, we made an energetic calculations with a methodology that is called MMGBSA. The results that we find in our investigation were that the 3 proteins were stable and also were able to establish interactions with the peptide that we simulate. We also measure the length of the interactions between the residues that are close to the motif, and we characterize them at an structural level as it shows in this image. Finally, with the energetics calculations, we were able to find an integrin that is higher in the energetic contribution, and we were able to locate the contribution that is highlighted in blue here that it was the biggest contribution to this result. With all of the above, we were able to conclude that both subunits share two sequence patterns and one geometric pattern, each of which are linked to the interaction with the RGD motif. The patterns identified in the alpha subunit tend to interact with the arginine residue of RGD, while those found in the beta tend to interact with aspartate. There are no notable energetic difference between the molecular docking results for the peptides that can be associated with a difference between both motifs. The most stable complex at the simulation level only generate interactions with the beta subunit as it was shown in this picture. The aspartate from the protein and arginine from the peptide interaction is established as the key to the anchoring of the RGD motif in the integrins. And finally, binding energies calculations were more favorable for the alpha 5 beta 1 integrin, described experimentally as being inhibited by the select peptide, establishing both peptides as alternatives. This was our investigation, and I really hope that you were able to enjoy my presentation.